Good afternoon, everyone. Lee Buckler, our CEO, was originally going to give this presentation, but unfortunately couldn't make it today. So I'm Kevin McElwee, and I'm actually the Chief Scientific Officer for RepliCel, although I do have an academic position at the University of British Columbia as well. But today I'm wearing my business hat or industrial hat. So just as a very brief introduction, RepliCel, it's a regenerative medicine company. And we're looking at a variety of different conditions and diseases where there's a deficit of cells, and we want to replace those cells. And so at the moment, we're looking at hair growth, but also things like tendons and also skin rejuvenation as well. So we have a number of different studies going on at the clinical stage, as well as some preclinical work as well. Now, before I go any further, I have to give a disclaimer. I'm sure you can read it all. Essentially, it says that we make no warranty or express or implied warranty about anything that's being presented today. Unfortunately, the lawyers insist that we include that. So the platform that RepliCel is using is fairly basic, regardless of what kind of condition we're trying to treat. We're using cells from the hair follicles in all instances, but we may be using different types of cells. So we take a biopsy from the back of the individual's scalp, and then we'll isolate our follicles and culture different cell types, depending on what we're trying to treat, and freeze those down, and then we send those frozen cells back to the clinic for injection, whether it's for skin or for hair or for tendons or for something else. So all of this actually came about through an original publication which I did when I was a postdoctoral fellow, along with Rolf Hoffman, who's the other co-founder of the company for RepliCel. And a long time ago now, back in 2003, we published this presentation where we used mice, and we looked at different cell types and whether we can induce hair growth in the mice. So this is actually a human hair follicle, but it was the same process with the mice as well, taking their whisker follicles and then isolating the relevant parts, in this case from the bulb and the lower part of the follicle. And so you're very familiar with hair follicles. You know these are the keratinocytes making the hair fiber. We're interested in the mesenchymal cells, the outer sheath of the hair follicle. And we split this up into different areas. There's the dermal papilla, which is the classic driving force for hair follicles that stimulate the hair growth, define the size of the hair follicle and the hair fiber being produced. And then there's also what we call the dermal sheath or non-bulbal dermal sheath, which certainly basically encases the follicle and is more of a structural factor. And then there's the dermal sheath cup, as we call it, around the bottom. Now, the papilla cells have been shown through many studies, way back actually to the 1950s, showing that they can actually induce growth or stimulate growth of hair follicles. You can actually produce brand new follicles using papilla cells. We were interested in the cups because papilla cells don't proliferate in the actual follicle itself. They have to be fed, it has to be supplied, and it's supplied by cells from the cup region. So we wanted to find out about these cells. So again, this is a human follicle, but the same process is done with mouse whisker follicles. We isolate the follicles, and we cut off the bulb, the little bottom bit, turn the cup region inside out to expose our papilla, and then we cut that off. And what we were examining was the cup cells out of this region, the dermal papilla cells, and then dermal sheath cells, the structural region cells. And if we culture these cells and then inject them into mouse ears, we get a variety of different results. With dermal papilla cells, we suddenly get hair growth. With the dermal sheath cup cells, we were also getting good hair growth as well. And then, unsurprisingly, the dermal sheath cells didn't do a lot. They certainly were in the ear, we could see them in the ear, but they weren't actually stimulating any hair growth, so it would seem. So this intrigued us considerably, and we found that there were a few subtle advantages, perhaps maybe, of using cup cells versus papilla cells. Particularly, you can see the angle of the hair growth looks more natural for a mouse ear, admittedly, but it looks more natural for a mouse ear versus the more sprouting effect that we saw with the dermal papilla cells. So this was one of the reasons why we liked the idea of using cup cells for promoting hair growth rather than the papilla cells. And if we label these cells and then implant them into the ears to see what happens, we got a couple of different presentations, both with papilla cells and with cup cells. With the cup cells, sometimes we could see follicles where all of the cells in the papilla were fluorescent, so that all of the cells we injected had gone in and produced this structure of the papilla, and also within the cup region as well. But then we found some follicles where there was a little bit of a mixture. So the papilla here is difficult to see, but it's actually fairly large, and only some of the cells are fluorescent. So these are the cells we injected. But there are other cells which are not fluorescent, so these must be host cells. And our interpretation of this presentation is that the cells that we've injected have migrated into the tiny little mouse ear hairs and actually made the follicles larger. And so the end result is that you get potentially a better distribution of hair because it's controlled by the little resident follicles. What we're doing is just enlarging the follicles. So our interest then is not so much to induce brand new hair follicle growth, but rather to expand, enlarge the miniaturized follicles potentially within patients. And so that was what we were focused on. So we started, after some years, a clinical trial. It was actually done in Georgia. That's Eastern Europe, Georgia, not US Georgia. Started back in 2010. And essentially, the process or the objective was to take people with thinning hair, so not fully bald, not extensively bald or long-term baldness, but rather to take those with thinning hair and to try and convert these vellus follicles into terminal follicles. And so what we do with the process then is, again, to take our little scalp biopsy. We take a 6 millimeter punch biopsy. That gives us about 20 follicles, give or take. And that's plenty for what we need to do. And then this is done in the clinic, of course. This will be shipped to us at RepliCel. We have a facility in Austria where we do the processing. And this is all done under GMP conditions. So the facility has to be approved by the local authorities and has to meet certain standards. And there's an awful lot of regulations around it. And it's all sterile environment and positive flow of air and so on. So we process the tissue samples and isolate pieces of tissue or cup tissues, put these into culture, allow the cells to start growing out, and then over time we start to see, obviously we get more cells, and we start to see the cells band together. They like to stick together. So it's a little bit different from the kind of standard culture you get where you just see the cells sort of in a flat row as they are in this location. A little later on they start to round up and collect themselves together. And we like to see that. That's a good marker of cells that are able to induce hair growth or promote hair growth. And we also do a lot of quality control, of course. Again, this is regulated by the local government authorities. We must have a number of factors that we look at within these cells to show that they are healthy, that they haven't mutated, and that they are expressing factors which are appropriate for cells from the hair follicle. So in this case, we look at alkaline phosphatase. There are many studies out there which have shown that alkaline phosphatase positive cells are good for stimulating hair growth. They induce brand new follicles or they'll stimulate resident follicles. So we want to see this within our cells. And these cells are actually from the phase 1 trial, one of the individuals in the phase 1 trial. So then we would send these cells back to the clinic to be injected. And in the clinical trial, of course, we're doing a very limited area. We're not doing the full scalp. Just a very small region was selected, and we had a tattoo mark to make sure we went back to the same region for monitoring and taking photos. And we used a special device which we built ourselves, which actually controls the amount of cells being injected and also the depth as well. The needle goes in at an angle and comes out at the angle and actually leaves a stream of cells along the needle as it pulls out. So we were actually spreading the cells out within the skin. And we did a very high dose of cells. And our knowledge now that we have now, we know that we actually gave too many cells. There's an upper limit to the number of cells that a piece of skin can actually accept. And you actually get a lot of cell death if you produce, or if you put in too many cells. So we were putting in a very large number of cells, but this was a phase 1 study, which is safety. So we had to prove that even a high number of cells is going to be safe. So that was the primary objective. And indeed, the results showed at 6 months and 24 months, and we're now up to 5 years with these patients, it does indeed seem to be safe. There are no significant side effects except for some pain at the point, at the time of the actual injection. And in terms of the results, although the study was not powered to actually look at the data and try and get statistical significance, we did look at the hair growth to see what we could see. And we got a variety of presentations looking at 6 months, looking in the individuals that we injected. So there were 7 patients who had more than 10% growth at the 6-month time point, and 10 patients that had over 5%. But you can see that there are indeed some patients who did not respond. We actually had some increase in hair loss. And unfortunately, some of these, at least 2 of these individuals, we think the reason is because it took a long time to actually ship the cells to the individuals in Georgia. They got held up at customs. And so they were actually outside of what's called the protocol. So they are not per protocol. So in the future, we have a time limit on delivery, which we must meet. Otherwise, these cells are not going to be so good for the patients. And then looking at 6 months, it may be a little bit difficult to see just by eyeballing it, but this is actually a 19% increase in density. This is at day 0 before the injection, and then 6 months later with a 19% increase. We also looked at thickness of the hair fibers. There is some thickness increase as well. I just wanted to briefly introduce some of the other things that we're trying to do with cells from, in this case, the sheath, from the non-bulbodermal sheath. These cells are much more structural. They produce a lot of collagen, much more so than the cells just sitting in the dermis. So we wanted to try and use these for places where there's a deficiency in collagen. And there are a number of different diseases and conditions where that occurs, but particularly we focused on tendinosis. So somebody has ripped their tendon. Tendons are just made of collagen, so we want to try and repair that. And then, more obviously, aging and damaged skin and old skin, which needs additional collagen. So in this case, we're taking the sheath and dissecting that off of the follicles and then putting that into culture and culturing the cells for about 4 weeks to 5 weeks. And then we take those cells, and then just as an aside, looking at the profile of these cells, looking at the marker expression. This is just one example of an individual we tested. So there are a number of markers that we try to look for. These are stem cell markers, and so the cells, these are MBDS in green, versus tendon cells versus something that was presented in a paper recently. So cells should be, if they're stem cells, positive for these markers, CD105, CD73, and CD90, and they should be negative for some of the others. So they have a pretty good profile in terms of being stem cell-like, but I wouldn't like to call them actual stem cells. I don't think they're quite there. I don't think they're quite at that level. But if you put the cells into culture, and if you put them into different culture media, they will do different things. So in a standard fibroblast media, they look like fibroblasts. If you put them into a media that promotes adipocytes, you start to get oil accumulation within the cells. They become more adipocyte-like. And then if you put them into media for bone cells, you start to get calcium deposition in these cells. So they're becoming more like chondrocytes. So these cells will do different things if you give them the right signals. But I wouldn't call them true stem cells. So we're going to call them multipotent cells. They will do some different things. So we're looking at these cells for tendon repair. And in somebody who's injured their tendons, what they do is they rip it apart. They create tears in their tendons. And basically you just get a dark space when you look at it under ultrasound. And what we want to do is to add cells in there that are going to produce collagen and repair that tendon. This was a previous study actually done with fibroblasts from the skin. And we followed that up with our current study. So with our cells, some of the tests that we did, for example, we looked at the cells, we put them into a matrix, and we stretched the matrix regularly while the cells were in culture. And if we do that, they become very stimulated to produce collagen. So this brown stain is very strong for collagen here after stretching, also for tenacin, which is relevant for tendons. So these cells do produce the right kind of factors for tendon repair. And so we had a phase 1 trial, very small trial with 10 people here in Vancouver. And very recently we presented some of the initial results, again at 6 months. So the individuals, some of them did very well. And there were a couple of people who almost eliminated their pain score, and they had significant improvement in the actual structure of the tendon as well. So people were doing, particularly in terms of pain severity, they were reducing their pain quite significantly by well over 50%. So we're very encouraged by this, and we will continue to do a much larger study. And then, of course, the more obvious thing to do with these cells is to try and do something on skin rejuvenation, particularly for aging skin. So you are very aware of what happens with aging skin, a loss of collagen, a loss of cells. The cells become senescent and less active. And so what we want to try and do is to add in new cells, which we're taking from the sheath of the hair follicles, and inject them into skin. And we did a trial in Germany with 15 women and 2 men, and then looked at them at the moment so far 6 months. We're still ongoing with this study, but we have data at 6 months. And we're taking biopsies from them as we go, and then looking for changes in gene expression. And what we see is that looking at some of the relevant genes for collagen 1 production, there's a significant increase, nearly 2-fold increase. Also for things like what's called a TIMP, which actually inhibits enzymes that break down collagen. That was also increased. Lumican is an extracellular matrix factor, which is relevant for maintaining collagen in bundles, and so this was also increased, and also TGF-beta was increased, which is something which stimulates the cells to produce collagen. So all these things are expressed, and they seem to be actually significantly expressed with, again, a single injection of these cells. And then finally, what the company has decided to do is to try and develop an automated injector device. And we need this for our cells. We need to be able to standardize how different people are injecting these cells. And the injector is electronic. There's a fairly complicated system inside with a number of different motors which control the depth the needles go to, how quickly they withdraw, the volume that gets injected, and how the injection occurs as the needles are being withdrawn. So you can actually have a triangle effect where you get more injected lower and then less and less as the needles pull out of the skin. And there are various different things that you can do with it. So this is going to be very useful for us looking at the cells. And we also have a number of different needle heads. So there's like a stamp sort of version, and then of course a single needle, then different needles in different formats for doing different things. And so the idea is that we can use this for our cells, but also this potential injector could be useful for other things like fillers, maybe for PRP. I'm not quite sure, but potentially it could be used for a variety of different things. So at the moment, that is the current focus of the company. And if you have any more details, of course, you're welcome to contact us. Thank you.